Good evening and good afternoon, everyone. My name is Suzanne Barnett, and I'm hosting your webinar for you today. As usual, for those of you that are on, we're still getting people coming in, but for those of you that are with us already, could you just give me an indication that you can hear me okay by putting your hand up for me? That would be most helpful. Thank you, Scott. Okay, just wait for a few hours. Thank you, Andrea. Okay, thank you. If you can't put your hand up, just type in the question box. That would be really helpful as well. So I'm going to assume that everyone can hear me and get on with the webinar. So once again, just to remind you, switch off any applications that you've got running in the background. It does make the quality of your webinar much better for you. Okay, we'll be aiming to be finished by 1 p.m. Not 1 p.m., 2 p.m. as usual, so within an hour and a half, and we will be allocating time at the end for questions. That said, if you do have any questions, you think of them, please type them into the question box as soon as they come up, and we will address them at the end. So our topic for today is our presentation 10, celiac disease, thyroid, eye health, and other immune factors. And our first presenter for today is Cynthia Smith. Over to you, Cynthia.
Thank you. Okay, so this particular module that we're doing today is a mixture of different types of SNPs that aren't really necessarily connected by one area. So we're going to be talking about celiac disease, thyroid function, eye health, and other immune factors. With the celiac, we'll be talking mostly about the HLA. With the thyroid, I think we're going to talk more about how the particular thyroid SNPs affect other organs. Eye health is the vitamin A deficiency. We have the SNPs on that variant report. And then when it comes to these other immune factors, it's a mixed basket. And what you'll see when you look at the variant reports, you'll look at a person's IgA SNPs, right? That's where you'll see some of these HLA SNPs as well as under the celiac section. You'll look at their FUT2 SNPs. And depending on what you see, if you see something or if they're presenting with some sort of autoimmune issues, you can go into this other immune factors and you can narrow down sometimes where their autoimmune condition might trend towards. Now I think Sterling's going to take us through some of the SNPs.
Hi, can everybody hear me? Hello, can everybody hear me? I guess you can.
Yes.
Okay, all right.
Sterling, we can hear you absolutely fine. Thank you.
Okay, all right. CTLA4. It is a common haplotype that is associated with celiac disease. CTLA4 RS231775 is a strong inherited risk characterized by dietary wheat, rye, and barley-induced T cell activation. CTLA4 plays a key role in regulating T lymphocyte-mediated inflammatory responses, and variants in the 3 region influence development of diabetes and thyroid disease as well as celiacs. HLA RS2858331. This HLA gene is related to celiac, diabetes, lupus, and rheumatoid arthritis. HLA-DQA1 gene provides instruction for making a protein that plays a critical role in the immune system. The HLA-DQA1 gene is part of a family of genes called human leukocyte antigen complex. The HLA complex helps the immune system distinguish the body's own proteins from proteins made by foreign invaders such as viruses and bacteria. At least 2 specific combinations of HLA gene variants have been found to increase the risk of developing celiac disease, a disorder in which inflammation damages the intestinal tract and other organs and tissues. One of these haplotypes, known as DQ2, is composed of the protein produced from HLA-DQA1 gene variants. DQA1 bound to the protein produced from HLA-DQB1 gene variants. The other haplotype is known as DQ8, is composed of a protein produced from HLA-DQA1 variants. DQA1 bound to the protein produced from the HLA-DQB1. The DQ2 and DQ8 haplotypes, which may occur separately or together, seem to increase the risk of an inappropriate immune response to protein gluten, which is found in wheat, rye, and barley. This immune system malfunction results in the damage to the body's organs and tissues that occurs in celiac disease. However, DQ2 and DQ8 haplotypes are also found in 30% of the general population, and only 3% of individuals with this haplotypes develop celiac disease. DQ2 is most common in Western Europe, North Africa. The HLA-DQA2 is most common in Western Europe, North Africa, and East Africa. Highest frequencies of the HLA-DQA2 are observed in parts of Spain and Ireland. This distribution correlates with the frequency of two of the most prevalent autoimmune diseases. There is also an increase in DQB1, which is part of DQA2, in Central Asia, peaking in Kazakhstan and slowly east to west, most predisposing factors for autoimmune disease. The DQ2 is encoded often by a haplotype associated with a large number of diseases. The haplotypes HLA, A1, B8, DR3, and DQ2 are associated with diseases in which HLA-DQ2 has suspect involvement. Direct involvement of DQ2 is certain in celiac disease. I hope I didn't confuse you guys. But that's it for that section right there. I'm Cynthia.
Okay, thank you, Sterling.
You're welcome.
So we're going to talk about celiac disease factors. I always get a kick out of the way you guys spell celiac in Australia. Okay, let's go to the next slide. So HLA stands for human leukocyte antigens, and one of the things that you'll see with these HLA SNPs is most of them, in fact I think all of them, are located on chromosome 6 and they sit close to each other. They're related to this list of conditions here, these autoimmune conditions, but I can tell you there are many, many more than this. I think the last count there were, the classification of autoimmune diseases was over 100. And what you're starting to see now in the population in general, at least here in the U.S., is autoimmune diseases are moving up in the stack of diseases that cause death, and they're inching up the ladder because of our dietary changes, the food chains. We're going to talk more about this with the Roundup, and I shouldn't say that, the glyphosates, how it's affecting our gut bacteria, and how when our gut bacteria are affected and we have inflammation, we then develop leaky gut, and then once you have leaky gut, these protein polypeptides cross that gut barrier, and depending on your predisposition and your genetics, it's possible to develop these autoimmune conditions. The priorities that I've seen, because I do a lot of Cyrex testing, I see a lot of Hashimoto's, a lot of autoimmune attacks on the pancreas, and so you'll see type 1 diabetes developing even later in life than was originally thought. It used to be associated with children. The other areas that are under attack that aren't really listed in this list are alpha-beta tubulin, synapsin, things that have to do with our nervous system and our brain function. So many of the people who are diagnosed with dementia, it's actually an autoimmune condition where their immune system is attacking parts of their nervous system. So in general, HLA genes provide instructions for making a group of related proteins known as human leukocyte antigen complex. The HLA complex helps the immune system distinguish the body's own proteins from proteins made by foreign invaders such as viruses and bacteria, but also by proteins which I refer to as polypeptides that are the result of food that has passed through, these polypeptides derived from proteins that have passed through the gut wall and also through the blood-brain barrier, because that's part of the picture too. We're going to talk more about this in the next class and about the blood-brain barrier and the fact that when somebody has an autoimmune condition, oftentimes if they don't know they have it and they continue to with their same eating habits, they can develop a leaky blood-brain barrier, and that's where we start to get into trouble with dementia-related autoimmune conditions. In humans, the MHC complex consists of more than 200 genes located closely together on chromosome 6. Genes in this complex are categorized into 3 basic groups: class 1, class 2, and class 3. Humans have 3 main MHC class 1 genes known as HLA-A, HLA-B, and HLA-C. The proteins produced from these genes are present on the surface of almost all cells. On the cell surface, these proteins are bound to protein fragments, peptides, that have been exported from within the cell. The MHC class 1 proteins display these peptides to the immune system. If the immune system recognizes the peptides as foreign, such as viral, bacterial, or polypeptides from protein foods, it responds by triggering the infected cell to self-destruct. And we're going to talk more about this, but there's something called molecular mimicry, where the immune system perceives, let's say, a gluten polypeptide, but because it looks very similar to thyroid tissue, you'll see people with leaky gut, autoimmune, Hashimoto's are very sensitive to gluten and should not be having gluten. And as time goes on, it's not just the gluten, that those gluten polypeptides will cross-react with other grain proteins. So really, once somebody's in this autoimmune, this autoimmune spectrum, most grains or many grains should be cut out as well. Okay, let's go to the next slide, please. So this slide is talking about celiac disease. Now, celiac disease is probably maybe 2% of the population. Gluten intolerance, on the other hand, is a much larger percentage of the population, and it's increasing. There's some work done by an Italian researcher named Alicia Fasano, who has done some of the best work in this area, and I would encourage you, if you're interested, to look up his papers. I also have some slides on my website where I've shown pictures of some of his papers and discussed his work in the celiac disease area. But celiac disease in general is an immune-mediated disease of the intestines triggered by gluten ingestion. And it's not just the gluten, it's the gluten, the gliadins, and within the gluten and gliadins there's subfractions. There's gluteomorphins, there's a whole bunch of subfractions that you can test for, but the bottom line is if you test positive for an immune reaction to just one subfraction, you might not have celiac disease, but you surely shouldn't be having gluten. 95% of celiac patients are HLA-DQ positive, 2 positive, and the rest are HLA-DQ8 positive. There's testing that they do to say that you definitely have celiac disease, and one of those, besides testing for these 2 markers, they can also do a biopsy and look at the tissue. One of the things that happens in all cases, though, with autoimmune is there should be a detection of a protein called zonulin or occlusion, which is what is instructing the tight junctions of the gut mucosa to open and allow these reactions to occur. And there's testing for that as well. Okay, let's go to the next slide, please. So this next slide is a nice little picture, and again, I've got other diagrams in a presentation that we taught in January that goes into much more detail. But genetic studies have confirmed the strong association to HLA and identified 39 non-HLA risk genes, mostly in the immune section. Over 50% of the disease-associated SNPs are correlated with gene expression. Most of the celiac disease-associated regions are shared with other immune-related diseases, as well as with metabolic, neurological traits or cancer. One thing I want to mention with celiac disease, many times it doesn't manifest as a gut problem where a person actually feels pain, and it doesn't necessarily manifest with extreme weight loss, which is what doctors have traditionally believed. It really identifies more with neurological issues. So with your clients who are having neurological issues, but they don't complain about their GI, I would still check to see if there's any autoimmunity going on, because really this is more of a neurological issue. Okay, let's go to the next slide. Okay, this slide just shows a little bit more detail about the MHC class 1 and class 2 antigen presenting pathways. So intracellular antigens such as virus or tumor antigens or food peptides are processed into peptides by the immunoproteasome, which is composed of multiple subunits including LMP2. The peptides are transported to the endoplasmic reticulum, where they are located into the groove of the MHC class 1 complex. And that class 1 complex is composed of a heavy chain and a beta 2 microglobulin portion. MHC class 1 complexes present the antigens on the surface of the cell to CD8 T cells. Proteins in the MHC class pathway that are encoded by genes regulated by the MHC class 1 transactivator, referred to as NOD-, LRR-, and CARD containing, actually those are all containing, 5, are shown in the yellow boxes. Antigens from the extracellular sources, such as bacterial antigens, are processed by endosomal enzymes into peptides. These peptides then bind to the groove of the MHC class 2 complex by displacing the class 2 associated invariant chain peptide called CLIP, which is derived from the MHC class 2 associated chain. HLA-DO, HLA-DM regulate the antigen loading process. The MHC class 2 complex presents antigens to the CD4 plus T cells. Anyway, so this is a very complicated slide, and it's hard to explain. These immune system slides are very difficult because there's much more involved, but it's a nice reference slide to have if you're trying to understand how the immune system reacts to these antigens. Now, Sterling discussed the CTLA4 and the HLA SNPs, and the next slide is just as a nice table where you can look at the risk allele and then what that allele has been associated with in the research. So, for example, for CTLA, there's an increased risk of lupus, autoimmune diabetes, which would be your type 1, colorectal cancer, and increased risk of asthma in children. Again, when you see asthma, you have to go back and look at the IgA SNPs as well. The HLA-DQA1, again, is associated with autoimmune issues, and then the DQA2 risk allele A, an increased risk of sclerosis. Now, Sterling's going to walk us through some additional SNPs.
All right. CTLA4, the human CTLA4 and CT28 genes are closely linked. The first evidence for association between the CD and CTLA4 genes in Graves' disease in white North Americans was reported back in 1995. Association of 3 polymorphic markers of the CTLA4 gene with the disease was demonstrated in most population-based investigations. So Graves' disease is highly related to CTLA4. Now, the FOXE1 gene, this gene functions as a thyroid transcription factor, which likely plays a crucial role in thyroid morphogenesis. Mutations in this gene are associated with congenital hypothyroidism and cleft palate with thyroid dysgenesis. The RS1867277 gene is associated with thyroid cancer. Cynthia?
Thank you, Sterling.
You're welcome.
So this next section we're going to be talking about thyroid factors, and this is a little bit unfamiliar territory for most of us. The FOXE, called the forkhead box E1 protein SNP. When I see this on somebody's report, I immediately look at other genetics as well, and I want to make sure also that that person has sufficient selenium and iodine. So we usually do that with a multiple, like a multi-mineral. Again, not too much iodine. I take a lot of people off of too much iodine. So this gene belongs to the forkhead family of transcription factors, which is characterized by a distinct forkhead domain. This gene functions as a thyroid transcription factor, which likely plays a crucial role in thyroid development. Mutations in this gene are often associated with congenital hypothyroidism, not Hashimoto's, cleft palate with thyroid degenesis. There are a couple of diseases associated with this. One is called Bramforth-Lazarus syndrome, or BLS, and it's characterized by thyroid anagenesis, cleft palate, and other things. But this disease is caused by the mutations affecting this particular gene. It's also associated with thyroid cancer, squamous cell epithelial, and sensory neuropathy type 1. Now this next slide is very complicated, and so I'm just going to mention that this FOXE1 pathway, there's been a lot of research done, and this particular SNPs in this particular area are associated with all of these things in these different boxes. They're affecting other genes as well. So, for example, craniofacial disorders in the bottom left, GI issues, neurons, lungs. So that slide is a nice a reference slide. Let me see if I want to say anything about this. Thyroid transcription factors, which are TTF. Let's see. Yeah, I'm not gonna go through this. This is too complicated. But the slide is there. There's various genes there you could look up if you're interested. Now here are the risk alleles associated with FOXE1 on the next slide. FOXE1 risk allele A is associated with increased risk of thyroid cancer. Studies have shown that the allele-dependent transcription regulation of FOXE1, specifically the risk allele A, increases the chance of thyroid cancer, but again, people need to be replete with their selenium and their iodine. Now Sterling's going to talk about the BCMO SNPs, which are the eye. We associate that with the eye pathway and vitamin A.
Okay, BCMO1, it symmetrically cleaves beta-carotene into two molecules of retinol. Hypercarotenemia and vitamin A deficiency is related to BCMO1. It is a disorder characterized by increased serum beta-carotene, decreased conversion of beta-carotene to vitamin A, and decreased serum vitamin A. I want to kind of share a little story I had one time when a lady called me. She was lucky enough to get one of the swab kits for her 18-month-old daughter. And she got the results back, and she was really upset because nobody could help her. She thought that her daughter was having issues breaking down foods, enzyme issues. And she called me and she was crying. She said, I know something's really, really wrong. And she's just not thriving right. And when we looked over her report, she had told me, she said, for instance, when my daughter eats carrots um, or yams, she told me that she would turn orange. And I said, I kind of think that this might be something that's a little simple as her not converting her beta carotene into vitamin A. And sure enough, when we went on her BCMO1, all three of them were red, every one of them. So she went and she brought her daughter to the doctor and they checked, and her beta carotene levels were through the roof, and her retinol was low, low, low. So they started lowering foods rich in beta carotene and started giving her more animal source vitamin A, and the little girl started getting well. It was amazing. So that's something to look at, because this can also affect more than eye. It can also affect thyroid and stuff, because guess what? When you don't have that animal source, that retinol A in your body, you're not going to be able to absorb iodine for the thyroid. Also, another thing, when you have a vegan and they're red in a few of these SNPs, know that there's probably a problem there with them as well. So that's really important to know. Cynthia, I guess you can kind of talk more about it now.
Okay, so as Sterling mentioned, this enzyme converts beta carotene into two molecules of vitamin A by cleaving the beta carotene. It's important for vision, embryonic development, cell development, skin membranes. And then, as Sterling mentioned, it causes hyperkerotonemia and vitamin A deficiency. But one thing I want to mention here, and this actually ties back to the immune SNPs, and Carolyn mentioned this last week, if somebody is showing on the autoimmune spectrum, they have something, they have Hashimoto's, if they have Hashimoto's, they usually have something else too, but any autoimmune spectrum issues, look very carefully at this enzyme, because if you're vitamin A depleted, then that makes the autoimmune condition even worse. So when we're looking at autoimmune, again, we look at IgA, FUT2, the autoimmune SNPs, the celiac, the HLA, and this particular SNP, the BCMO SNP, because we have to ensure appropriate amounts of vitamin A, or that autoimmune condition can get much worse. Now the next slide is a nice little diagram that shows the metabolism of carotenoids and retinoids, and that they begin in the intestinal lumen, where provitamin A carotenoids and retinoid molecules are absorbed. Retinol esters are transported to the liver via the lymphatic system, while retinol is transported through the bloodstream before delivery to the target cells, such as the retina. Now the next slide is the BCMO1 SNP functions with the risk allele, and you can see here that for the second RS number, the risk allele is T, and it results in increased plasma and retinal concentrations of lutein, and also reduced catalytic activity. So the bottom line is, if somebody's ingesting, let's say, a vitamin, a lot of these vitamins are combinations of beta carotene and retinol, vitamin A. People with SNPs here, you don't want to give them vitamins with beta carotene in it, especially if they're homozygous, because they're not going to be converting at all to the vitamin A. So be careful of that. And as Sterling mentioned, if you've got somebody with like 3 homozygous reds, and we're only looking at 3, those people might be sensitive to foods that are high in beta carotene. And well, I'm not going to go there. But anyway, just be cognizant of these multivitamins with beta carotene if somebody has BCMO1 SNPs. Now, Sterling's going to go through the immune factor SNPs. And like I mentioned earlier, this is kind of a grab bag of immune SNPs. And depending on other SNPs associated with the immune system, these particular SNPs will kind of guide you into what area to look in for additional autoimmune conditions if they've presented with one already. Okay, go ahead, Sterling.
Okay, 4Q27, I'm going to leave how it works for Carolyn, as she better understands how it actually works, but I'm going to talk about diseases related to it. Now, parasites and mutations at 4Q27 play a role in inflammatory disease. Diseases related to 4Q27 are rheumatoid arthritis, celiac, type 1 diabetes, ulcerative colitis, inflammatory bowel disease, prostate cancer, giant cell arteritis, juvenile idiopathic arthritis, autoimmune inflammatory diseases, multiple sclerosis, Crohn's disease, cholestatic liver diseases, and lupus. Now, APOE, we've been over APOE before. Now, APOEs, they really need to keep an eye on their fatty acids, and especially the omega-3 to 6 ratios. They have a much harder time detoxing metals from the brain. So people with mercury amalgam fillings, and you see APOE, you wouldn't want to immediately have them removed. You would want to make sure that their diet's well balanced, that they're kind of taking care of this omega issue, fatty issue that a lot of APOEs have before even considering, and methylation's working, considering removing those mercury amalgams. And they would want to definitely use a biodentist, preferably trained by somebody like Al Huggins. Now, Stephen Gundry, he's an MD and cardiovascular surgeon. He calls APOE 4 4s and 3 4s grain brains. And he has hundreds of peer-reviewed research articles out there on APOE. This is a pretty brilliant man. He also invented the device when you're in heart surgery that it keeps your heart beating. The man really is brilliant. He has a lot of research there on the vascular system and how APOE works. He says that even consuming a cow that has been fed grain is bad for people with APOE 4s. Now, use caution with ALA on people with APOE mutations and mercury amalgam fillings. I've actually had a researcher come in and talk about that, you know, when the mercury is not detoxing in the brain, which ALA is actually good for somebody with APOE, but when they have something like mercury in their mouth and they don't have their omegas and stuff balanced, and they take ALA, that they can redistribute and bounce that mercury all back and forth in their brain. So that's another thing to look at to make sure things are working and mercury amalgams are possibly properly removed before ALA is given to them, because actually it's good for APOEs, but not when they have the mercury in their mouth. Also, methyl B12 tends to do this to APOEs. It'll move the metals in the brain back and forth. So if you have an APOE 4 and they need B12, methyl B12 might not be the greatest thing for them. You want to make sure the omega-3s and 6s are back in balance as well. They really can distribute and bounce back and forth in them and cause a lot of problems. I met a lady who was loaded with mercury amalgams and is a 4-4, and knowing that her doctor knew that ALA was actually a good supplement for these people, but didn't know the research between having a mouth full of mercury and APOE 4. And when she took a low-dose ALA, she was in the hospital for a week. She didn't even know who she was. She had amnesia. So now ATG16L1. This gene is located in chromosome 2, and it codes a protein involved in the sorry about this. I'm getting to the next page. In the formation of autophagosomes during autophagy. Autophagy is a cytoplasmic process that keeps a cell stable. Researchers found that the variant of ATG16L1 was associated with a higher risk for Crohn's disease and ulcerative colitis. For child-onset inflammatory bowel disease, this variant was related to Crohn's disease, but not ulcerative colitis. These findings suggest that a role for autophagy in the development of inflammatory bowel disease And the exact role of ATGs they really don't quite understand how it plays a role in IBD, but they know it does play a role in inflammatory bowel disease. Now, GSDMB, that intestinal biopsies have shown that GSDMB is highly related to inflammatory bowel disease. So people with GSDMB mutations have a higher risk of IBD. Also, it plays a role in epithelial cells. It is also responsible for the health of gastric chief cells and colonic surface mucus cells in the basal region of neuroendocrine cells. So mutations at GSDMB can cause inflammation in organs because of this. Now, HLA-DRB1 is associated with inflammation in multiple sclerosis, the gut, rheumatoid arthritis, type 1 diabetes, Crohn's disease, ischemic heart disease. Poor glutamic acid breakdown has been associated with HLA-DRB1 genes and the rubella virus with T cell involvement. DRB1 is highly associated with latex sensitivity. So if you see somebody with HLA-DRB1 genes, they might not do good laying on one of these new mattresses that they have at all the hotel rooms that are loaded with latex in them. Might not be a good idea for them. People with HLA-DRB1 mutations tend to have higher IgE serum levels. It is also associated with interleukin-4 secretion. IL-13, interleukin-13, has cytokine involvement. It inhibits inflammatory cytokine production. It also synergizes with interleukin-2 in regulating interferon gamma synthesis. It may be critical in regulating inflammatory and immune responses. IL-4R, interleukin-4 receptor. For both interleukin-4 and interleukin-13, the IL-4, IL-13 responses are involved in regulating IgE production and chemokine and mucus production at sites of allergic inflammation in certain cell types. And it can signal through activation of insulin receptor substrates. Allelic variants in IL-4 or A are associated with susceptibility to atopy and immunological condition that can lead to clinical symptoms such as allergic rhinitis, sinusitis, asthma, and eczema. IL-4A or A variants are associated with cedar pollen sensitization. Individuals develop Japanese cedar pollenosis with increased exposure to cedar pollen. Japanese cedar pollenosis is a type 1 allergic disease with ocular and nasal symptoms that develop on contact with Japanese cedar pollen. These symptoms, which occur seasonally each year, are typical features of allergic rhinitis, such as sneezing, excessive nasal secretion, nasal congestion, and conjunctival itching. IL-5, interleukin-5, it is a protein coding gene. Diseases associated with IL-5 include gastroenteritis and eosinophilic gastroenteritis. Among its related pathways are TGF-beta pathway and TGF-beta pathway. The annotations related to this gene are cytokine activity and interleukin-5 receptor binding. Interleukin-5 is a factor that induces terminal differentiation of late developing B cells to immunoglobulin secreting cells. The pharmaceutical industry is in the process of making interleukin-5 blockers for allergic responses. So they found that this is a big deal, especially in mast cell people. So there's none out there, even in trials, I don't think yet, but they've been in the process of figuring out how to make interleukin-5 blockers. Like they have interleukin-13 blockers for asthmatics now, that they just haven't quite developed the IL-5 blocker yet. MEFV, Mediterranean fever, this gene, really important, and not a lot of people know about it unless you're in an Eastern European country. Doctors are very good at it there, but unfortunately, they're not too well versed in other regions. Now, this you will see in Ashkenazi Jews, people from Turkey, people from Greece, lots of problems with this, and also Asian coastal regions. And this gene is related to familial Mediterranean fever, autosomal recessive. Now, it's a hereditary periodic fever syndrome characterized by recurrent episodic fever, serosal inflammation, and pain in the abdomen, chest, joints. It is frequently complicated by reactive amyloidosis, which leads to renal failure and can be prophylactically treated with something called colchicine. It's called colchicine. So Mediterranean fever, there's two different types. The autosomal dominant is a hereditary periodic fever syndrome characterized by the periodic fever, serosal inflammation, and pain in the abdomen and chest and joints, as seen also in the autosomal recessive form of the disease. So it is associated with reactive renal amyloidosis and is characterized by colchicine unresponsiveness. So the recessive, they can take the medication and they fare better. The dominant, they're going to take the medicine and they're not going to respond as well. These people usually end up on dialysis and they end up with liver transplants. Now, someone with chronic inflammation and Mediterranean fever genes should be checked for elevated IgA, elevated IgG, and elevated IgM serum levels. It's really important. We usually, when we see IgA and IgG, we see low. You ever get somebody with elevated immunoglobulin markers, that's A, G, or M, and you see Mediterranean fever SNPs, you get them to the nearest facility to get medical help immediately. That's how serious this can be. Now, they're serious, and if you do have, like I just repeated, you have a client. Now, if they have inflammation, kidney pain, jaundice, or other signs of liver issues, chronic fever, and even hallucination, get them the proper testing and emergency medical treatment ASAP. So, and you will see chronically ill people with a SNP here. You want to rule out that you don't think that they're, you haven't seen any labs yet, and you do check IgA, IgM, IgG. You see any of them elevated, and you see someone with a Mediterranean fever SNP, get them help immediately. And that is Ashkenazi Jews, Turkish, Greek, and Asians. MTHFR genes, some if left untreated do require the liver transplants, and if not caught in time, they can die of liver and kidney failure. I've even seen people have hallucinations from this. I have somebody that's close to me that is an alcoholic. People with Mediterranean fever genes, and especially the chronically ill ones, have no business touching alcohol. This person's waiting for a liver transplant, by the way. All right, step 4. STAT4 is involved in interleukin-12 signaling. It acts with IRF5 to increase. It acts with IRF5 and increases the risk of lupus. So STAT4 and IRF5 together, mass inflammation in really sick people, and it is related to lupus, rheumatoid arthritis, antiphospholipid syndrome, systemic sclerosis, and juvenile idiopathic arthritis. And antiphospholipid syndrome, believe it or not, a lot of those people do have lupus. So if you ever have somebody with lupus, you want to check them for antiphospholipid syndrome. If you ever have somebody with antiphospholipid syndrome, you want to check them for lupus. TNF, tumor necrosis factor. TNF was thought to be produced primarily by macrophages, but it is produced also by a broad variety of cell types, including lymphoid cells, mast cells, endothelial cells, cardiac myocytes, and adipose tissue, fibroblasts, and neurons. This one is big inflammation, so a lot of people with mast cell, you'll see TNF SNPs on them. Large amounts of TNF are released in response to lipopolysaccharide and other bacterial products in interleukin-1. In the skin, mast cells appear to be the predominant source of preformed tumor necrosis factor, which can be released upon inflammatory stimulus. It is a number of actions on various organ systems, generally together with interleukin-1 and interleukin-6. So we hope to get more of those interleukin genes out on our autoimmune report, so you guys can look at, you know, how tumor necrosis factor does play with the interleukin genes, because you'll see a lot of inflammation in those people, sometimes arthritis, big time. So TYR, that gene, the TYR gene encodes the tyrosinase protein, a key enzyme in the synthesis of the melanin pigment. Variations in the TYR gene sequence have been linked to numerous pigmentation phenomena in mammals. Now, FUT2. We didn't put FUT2 on here, and I thought it was really important that we add this in. So maybe we can add a note to you guys on the RSIDs I'm getting ready to talk about. And if you have a pen and paper, you might want to grab it down now and write it down. But FUT2, there are three that work together, and I'm going to give you the three RSIDs. RS492602, RS601338, RS602662. Now, all three of those together, and then there's another one that works alone. It's RS1047781. Now, when you see these heterozygous, no big deal. But when you see that first group of three, homozygous, or that last one I talked about, homozygous, these people cannot produce H antigen in the gut. And they also can't produce it in their mouth. So you'll see them with earlier tooth decay. It's good for them sometimes to use like a probiotic oral rinse in their mouth a few times a day. They will have a lot more gut issues and inflammation. And I don't know who, I think maybe Cynthia might at the end tell you a little more about it, or maybe even Carolyn. But it's very important to look at that when you're looking at someone, especially when you see that they have a lot of IgA issues. Along with that, there could be a lot of immune issues and gut issues. So Carolyn, I think I'm done. Ready for you.
Thank you, Sterling. Great job. All right, so I'm going to just take you through these in a little bit more detail. And first of all, we're going to talk about the 4Q27 region. So think of this as it really encompasses four key genes, and they are KIAA1109, TENR, interleukin 2, and in fact, I'll go to the next slide because it's written there, and interleukin 21. And they're all located on chromosome 4q27. That's why it's called the 4q27 region. And there are probably 5 key RSIDs that we look at, and certainly that the research looked at. And the one that we've got on the variant report, which is the RS6822844, is the key one that we associate with rheumatoid arthritis. And indeed, the whole region has been linked with autoimmune disease, not only celiac disease, but rheumatoid arthritis and type 1 diabetes. And Cynthia Sterling at the end is going to just make mention of the different colours on the variant report and what they mean. And this whole region has been studied in inflammatory bowel diseases, as Sterling said, like Crohn's and ulcerative colitis. And recent evidence is also showing some sort of link with psoriasis and psoriatic arthritis. The interleukin 2 and interleukin 21, I guess, are the most important components for me as part of this region, because they belong to obviously our type 1 cytokine family, and they have multiple functions in immune cells. And as we know, these two genes really have a genetic impact on autoimmunity. It's the interleukin 2 that has effects on many cell types, especially our T lymphocytes. And so a major function of interleukin 2 is obviously the promotion and proliferation and expansion of our CD4 plus and CD8 plus T cells. And they also help induce other cytokines. So really important role. We also know that interleukin-2 plays a role in CD4 plus and CD25 Treg cells, and these also have an important part to play in autoimmunity. There's a lot of studies that link this with rheumatoid arthritis. And as far as the interleukin-21, that's involved in both cell-mediated and humoral responses and has an effect on a variety of immune and non-immune cells. It also has a crucial role in the regulating of antibody production, particularly our Th17 cell biology. And in rheumatoid arthritis, with synovial fluid and tissue enhanced inflammatory response, you can see that with an increase in interleukin-21 and an increase in interleukin-21 receptors, that's what's causing that enhanced inflammatory response. And a crucial role of interleukin-21 is also autoimmune pancreatic beta cell destruction. They've seen studies in mice that show that this interleukin-21 is having a major effect on beta cell production. So that's quite interesting as well. Okay, so the APOE, we've seen this before, and as Sterling said, we've talked about these genes that make instructions for apolipoprotein E, and really they combine with our fats, as we've said before, to create lipoproteins, which are responsible primarily for the packaging up of cholesterol and other fats that are then carried through the bloodstream. And it's the APOE4, which is the largest known genetic risk factor for sporadic Alzheimer's disease. And it has major implications in normal cognitive aging decline. And I guess why we're talking about it with regards to the immune function is that both the APOE3 and 4 are involved in all major cellular amyloid beta clearance pathways. So that's why it's in this group. ATG16L1. So as Sterling said, it's crucial for autophagy, and it's part of a larger protein group that really, what happens in autophagy is that the cells will use this process to recycle any worn out cell parts and break down proteins that they don't need anymore. So and the autophagy also plays a really important role in cell death, so apoptosis. And you'll see a lot of genes in this pathway, like tumor necrosis factor, that has important implications on apoptosis. So if we're not killing off cells that need to be and getting rid of this harmful bacteria that perhaps stay in that would otherwise be destroyed, then we're going to have problems with the immune response. We're going to have problems with inflammation, and that's why one of the mechanisms behind its links with Crohn's disease. And I think predominantly they've seen this connection with Crohn's disease in white populations. So you can see here that the genes are linked with rheumatoid arthritis for the 4Q27 region. APOE, we know the risk with Alzheimer's and mild cognitive impairment, and the ATG16L1 also with Crohn's disease. So GSDM, this really is a Gasdermin domain containing protein family. Again, another one that has a key role in the regulation of apoptosis in epithelial cells. And that's why this has a link with cancer. In this particular study, they looked at how the GSDMB pathway has a role in epithelial barrier function, and particularly the genes in the airways that express the parts of the epithelial may enhance vulnerability to environmental insults. And so you get a disturbance in the epithelial repair, and that then will cause issues around epithelial barrier function and obviously immune processes. And this was how it's specifically related to asthma. HLA-DRB1 pathway. Its official name is Major Histocompatibility Complex Class 2, or DR-beta1. And as this is probably, as Sterling said, one of the most critical for the immune function, because it's a part of our leukocyte antigen, or our HLA complex, which helps our immune system distinguish the body's own proteins from proteins made by foreign invaders like viruses and bacteria. And the HLA-DRB1 gene belongs to a group of our, as we said, our major histocompatibility complex, the class 2, and these present proteins attached to protein fragments outside the cell. The MHC class 2 proteins then display these peptides to the immune system. The immune system recognizes them as, say, foreign, such as a virus or a bacteria, and then it triggers a response to attack the invading virus. So this protein that the HLA-DRB1 gene produces is called the beta chain, and it attaches to the alpha chain, and that's how it forms the HLA-DRA gene. And together, the HLA-DRB1 and the HLA-DRA form a complex that is called the HLA-DR. So as far as the let's see if I've got the HLA-DRB1 gene, it's associated with an increased risk of developing autoimmune Addison's disease. This is when the immune system malfunctions and attacks the adrenals. We haven't got specific research on that. It also is associated with an increased risk of developing multiple sclerosis, because it's obviously involved in the immune system, and the changes they believe is related to the autoimmune response and inflammation that will then damage nerves and the myelin sheath. It's been linked obviously with the rheumatoid arthritis and also with type 1 diabetes. All the interleukin genes. So we know that all the genes in the interleukin family will provide instructions for making our interleukins and our interleukin receptors. And these are all part of our cytokine group. And the cytokines are really what control our cell-to-cell communication. So think of the functions are really immunity, inflammation, formation of blood cells. So crucially important when it comes to our immune system. And the interleukins, don't forget, are also produced by T lymphocytes, B lymphocytes, and they have many functions that promote growth of immune cells and help regulate the immune system. There's about 33 different interleukins in humans, and they have been associated with many diseases, and you can see a list of them there: ankylosing spondylitis, Crohn's disease, idiopathic inflammatory myopathy, multiple sclerosis, psoriatic arthritis, and X-linked severe combined immunodeficiency. So most of these interleukins will exert their effect by having an effect on the interleukin receptors, and these are embedded in the outer membrane. And it's when an interleukin attaches to its receptor, that's when we get this trigger of our chemical signals. And in this section, we're specifically talking about interleukin 5 and 13. So I thought this was probably a good diagram to just give us, we can see that Th2 cells upon antigen stimulation produce large amounts of interleukin 4, interleukin 5 and interleukin 13, and that Th1 cells produce our interferon gamma, and that Th17 produce interleukin 17, 21 and 22. But there's interesting research coming out about it that it's not so much regimented as we've seen in the past in studies. And so some of these new studies are coming out which are really interesting that look at several distinct memory type Th2 cells, and they produce large amounts of interleukin 5, interleukin 13, 17 and interferon gamma in addition to their interleukin 4 and 13. And it's believed that they play a really important role against infection and inflammation. And our memory CD4 cells play such a crucial role in providing long-term antigen-specific protective immunity. And so recent studies are showing too that these memory Th2 cells produce the interleukin 5, and they then produce memory Th2, sort of almost like a subset of cells, and that will induce an allergic inflammation. And you can see here that chronic inflammation caused by these pathogenic memory type 2 T helper cells, which they're calling them, in the airways and skin. And you get a chemokine receptor 4 being expressed, which produces interleukin 5, the memory Th2 cell, and that's when we get that infiltrates into the airway where they react with antigens and produce Th2 cytokines that lead to our chronic eosinophilic inflammation. Complicated story, but if you're interested, I would really encourage you to read this paper. It was very enlightening and really good. But again, as Cynthia said, we're getting into a very complicated pathway, these immune cells. I mean, we could probably do 13 webinars just on our immune cells. Not that we will, by the way, but we could. And so there is, you can see with the interleukins 13, there is an increased risk of allergic rhinitis, psoriatic arthritis, also associated with DNA methylation interactions in asthma, and has therefore an effect on our lung function. And the IL4R, which I didn't talk about specifically, but because there was no real studies that showed part of this inflammatory pathway, but it was associated with familial high cholesterol. And interleukin 5, as Sterling said, is linked with an increased risk of Graves' disease. So Mediterranean fever, I think Sterling and Cynthia know a lot more about this than me, but really it's interesting because I didn't know that it's the most common periodic fever syndrome in the world, particularly amongst Eastern Mediterraneans. And it's this MEFV gene that encodes for something which is called pyron or marinostrin. And the pyron is really there to keep inflammation processes under control. So when inflammation occurs and the immune system sends its signal out and we get white blood cells to the site of injury, we get repair. However, when pyron is produced in certain white blood cells, particularly neutrophils, eosinophils, and monocytes, it actually plays a role in inflammation and in helping to fight this infection. So they believe that with pyron there, you get this direct migration of white blood cells to the site of inflammation, and that stops and certainly controls the inflammatory response. And it interacts with other molecules involved in fighting infection and in the inflammatory response. So it's interesting that really it's helping us regulate inflammation, and because it interacts with a lot of these other inflammatory markers. There's more than 80 MTHFR genes, and a few mutations they say completely delete the pyrin, and others just reduce it. And I think that's probably the key thing, is that if you're getting a reduction, you're getting a reduction in the ability to control this inflammation. And here, this really just shows us that the MTHFR is associated with this elevated pyron. And you can see there the little star shows that the pyron is being reduced, and therefore it has a direct effect on the ability for the interleukin. Well, it actually increases the interleukin 1. So the other immune, you can see there mostly there are a few key genes that are involved. The key ones that we've found that are linked with this are E148Q, M6801, M6941, M694V, and V726A. V726A. And these 1, 2, 3, 4, 5 are estimated to really cause 70% of the Mediterranean fever issues. And I've highlighted them on these particular slides. So STAT4, it's a gene that provides instructions for a protein that acts as a transcription factor. So what that means is it's going to attach or bind to specific regions of DNA, and it will help control the activity of that particular gene. And it's turned on by cytokines, and when activated, the STAT4 protein increases the activity of those genes, particularly Th1. And so that's why we're seeing this link to several autoimmune type conditions like SLE, RA, Sjögren's predominantly. And here you can see some of the STAT molecules have been identified as playing a role in Th1 and Th2 cell polarization. So STAT4 is activated in response to interleukin 12 via the interleukin 12 receptor, and that then controls the upregulation of interferon gamma, leading to a Th1 increase. So tumor necrosis factor, as Sterling said, and as we've said before, it's a pro-inflammatory cytokine, and it's involved in cell proliferation, differentiation, again, apoptosis. So we're seeing a lot of these immune genes being involved in apoptosis. So the ability of the cell to kill off genes that may be harmful, or kill off viruses and bacteria that may be harmful, is going to affect the immune process. And therefore, all these genes that are involved in our apoptosis, we've got to consider potentially as having a major impact on not only autoimmune but possibly cancer. And the TNF alpha is involved, as we know, as part of our antigen-specific IgE, and as a result, it's been involved with allergies. And there is quite a significant association between IgE and the RS361525. And on our sheet, that is the, which one is that? That is the, we haven't got it on our sheet, but the, yes we have, it's the TNF238 is RS361525. So that's been associated with high serum IgE levels. particularly in females. And there's been a few studies that look at this TNF 308 promoter polymorphism and asthma. And there seems to be a very clear association between this SNP and overall susceptibility to asthma. And that's the RS1800629. And I think you saw that on the other. It'll be coming up in a sec, so I'll show you those when we get to them. And now this one is a really interesting one, and looking into this a little bit more, I sort of thought I could really get my teeth into researching this and the effect of this particular gene a lot more, because as Sterling said, this TYR gene really provides instructions for making tyrosinase, and as you know, that's located in melanocytes. So it has a direct effect on melanin, which obviously gives our skin, hair, and eyes their colour. So if there is you can see it's involved in that first step in melanin production. And it converts tyrosine to dopaquinone. And so it will then have a series of chemical reactions that puts melanin in the skin, the hair follicles, and the coloured part of the eye. So there's been a few studies that have linked this with it's oculocutaneous albinism type 1, and that's obviously the much more, I guess, it's the one that we see a lot more with abnormal production of melanin, so really loss of colouring in the hair, skin, and eyes. And that's a lot more serious in a way that these people just don't have any colour. But there is a second version that obviously isn't as I don't want to say serious because it's not serious, but it's not it doesn't seem to have as big an effect, but it will allow people to sort of have a less of a change in colour of the skin, and predominantly this is a decrease in activity of tyrosinase. So my thing was I really wanted to see, well, what are the other implications of this as far as tyrosine goes? What effect might this pathway have on dopamine, and what effect might it have on neurotransmitters? I don't know the answer, but it would be something interesting to pursue. So if we look at these STAT4, as we said, autoimmune conditions, SLE, rheumatoid arthritis, autoimmune thyroid disease, TNF 238, definitely associated with allergic rhinitis, an increase in IgE, TNF 308, there was the link with asthma and psoriatic eye disease. And TYR, no specific research found, but I'm definitely going to go back and look at the mechanism of what else we might find in that pathway. So some of the things, and we'll talk about this more in question time, but you mean definitely with the immune system, as Cynthia said, vitamin A is really key, vitamin D, obviously, all our fat-soluble vitamins are important. So again, we're not looking at anything in isolation, but we're certainly looking at this group with many other parts of our pathway. And when you see people present with autoimmune disease of any sort, it's important to look at all of these. All right, so that's my part done, and I think we'll hand over to see if there's any questions, Suzanne.
Okay, great presentation again, and yes, we do have some questions for everyone, so let's actually see what we've got here. So it's not a huge amount of questions, so please take the opportunity to type your questions in for us right now. So the first one that I need to ask you is that, let me see, okay, Sterling, you mentioned an expert earlier on with an Italian sounding name. Could we have the spelling of that, please?
An expert was that Stephen Gundry for APOE.
No, I think it was Cynthia talking about Professor Alessandro Fasano.
Oh, yeah.
Oh,
Yep.
right.
I'll type it into the chat
Could you?
section. Yeah.
That'd be great. Okay, so whilst you're typing that in, I've got a question here for you. ALA is alpha lipoic acid or LA, sorry, let me say that again. ALA is alpha lipoic acid or ALA fatty acid.
Alpha lipoic acid.
Okay, and sorry guys, that's all the questions I've got.
Okay,
So,
I wanted to add something on that
please do.
APOE because it just struck me. I had come across a lot of studies when Carolyn had been talking about TNF. I came across for the past couple of years several studies with APOE4 and tumor necrosis factor together. Very, very, very bad. If you've got to get inflammation under control, leaky gut healed. These people are even at tenfold more increase of getting Alzheimer's with TNF and APOE4 if they're living an unhealthy lifestyle. They're more than likely going to develop Alzheimer's if they live long enough. If those two genes are together and they're not doing anything health conscious, they're going to have problems. I think that's real important. And I wanted to add, do not ever think that somebody with APOE doesn't need alpha lipoic acid, because they really do, and it helps them. But if you see that they're very unhealthy and they do have the mercury amalgam still in their mouth, you want to make sure that you get them a little bit healthier and start getting the mercury amalgams removed before giving ALA.
Okay, I've got a question here from Micheline. Any recommendations beyond appropriate O3-O6 balance for fatty acids on APOE, E4 and E3?
Cynthia, do you want to take that one?
Cynthia, I think you're pretty good at that one.
For APO, APO4s,
Yeah.
APO4s. So APO3 is like the middle of the road. APO4 has risks and APO2 has risks. And so when you have combinations of like a 3 and a 4, a 3 and a 2, or a 2 and a 4, that's a pretty bad one. You have to be careful. And again, if you're talking about fats, there's some basic things besides balancing the 3 and the 6. Also make sure that that person is not eating fats like canola oil, fats that are derived from GMO foods. Obviously, trans fats are bad, but just in general, their fat balance has to be carefully monitored. And the NutriEval test has a whole section where you can look at their 6, their 9, their 3s, and all of these other fats, and also their enzymes. Like the 6-dehydrogenase, the enzymes that convert between the various fats. So I would monitor them pretty closely, and then of course take out the bad fats.
Okay, thank you. Can you, Carolyn, can you flick on to the last slide again, please? Sharon's asking to see that again.
Okay. That one?
Okay, is that the one that you wanted, Sharon? You can just put your hand up. Yep, thank you. Okay, and now we don't have any more questions, so is there anything else that any of you want to add?
Yeah, I would like to. I would like to, because Carolyn mentioned it and I forgot to mention it. The 4Q27, all right, we put the risk allele as T, because we see a lot more with the risk allele T. So if you see somebody green with 4Q27, that means their risk allele, they're G, they're GG. But a lot of the reports on celiac disease is related to the risk allele being G, but the majority of the reports for all the other inflammation is related to T. That's why we have T out there as a risk allele. So that's one of those genes. If you have it one way, there's a problem. If you have it another way, there's a problem. So on that 4Q27, we do have on the variant report. I wanted to tell you all about that.
Okay, and whilst you were just saying that, we've got another question that's coming. Is it true that FUT2 affects B12 and bifidobacteria?
Yes. Well, Sterling, you might
Cynthia?
want to say more. Oh, Cynthia, you can answer more. But yes,
She's pretty
basically,
good at that one.
yeah.
Yeah, if you have FUT2 SNPs and you do a quick research, Google, it'll talk about B12, but it really goes much deeper than that, and it has to do with the blood group antigens that are embedded in your gut mucosa, which are, of course, dictated by your blood type, but also, I don't know if you remember that book about Eat Right for Your Blood Type. There's something to that, because certain people, because of their FUT2 status, just are not, they just don't tolerate eating certain foods. And so, for example, I'll take the example, this is kind of an aside, like latex. So somebody has a latex allergy, and they have FUT2 SNPs. That person might want to also avoid latex foods like avocado, mango. So it gets a little bit more complicated. FUT2 homozygous SNPs are usually meaningful and are usually expressing if people are eating the wrong kinds of foods. And it might not necessarily be as simple as gluten and cow dairy. It could be, much more complicated based on their FUT2 status and some of their other immune SNPs. Thyrex testing offers a new array. It actually came out in February, where they look at foods that are well beyond what most of these testing companies do to identify foods that cause immune response. So I'll just put that out there. I think they test 210 foods beyond what a lot of these other companies test for.
All right, I wanted to add something to that. Jacqueline Greenfield is one of the authors of the Eat to Your Blood Type, and she's a friend of mine. She volunteers on my forum at MTHFRsupport.com. We have a big forum there, and she works hand in hand with all of the blood type people. Her husband and her are some of the writers on Eat Right to Your Blood Type, and you can actually find her under the Find a Practitioner on my website in the UK and also in Arizona. She lives half the year in the UK, half the year she has a practice in Arizona. And she loves when people contact her about anything about FUT2. And she's really big on, the foods and the different blood types for FUT2s because they are very sensitive. So when you go on those blood type diets and you see secretor and non-secretor, the homozygous FUT2s are the non-secretors.
Okay,
Yeah.
excellent. Is there anything else that you want to add, Carolyn?
I haven't got my variant report in front of me, but it's the middle FUT2 RSID that does affect the bifidobacteria, and the mechanism is that some microbes can use blood group antigens or glycans found in, say, ABO antigens, and it's the ABO blood group antigens that aren't in, a lot of people don't have it. And as Sterling just said, it's the non-secretor blood group. If they don't have a functional FUT2 gene, then they have a problem with these blood group antigens. And so they don't have the ABO antigens in their secretions and mucosa. And that's why they have this problem with, particularly with the bifido. But I can't remember the RSID. It's the middle one, Sterling. Okay.
We've got another question. Which number on Cyrex?
What I usually do is I run the array 3, the 4, and the 10. The array 4 is, the array 3 are the fractions of gluten, so sometimes you have to prove to people that they have an issue with gluten. So I'll use the array 3. The array 4 is a list of foods that people tend to migrate to when they go gluten-free. So that includes a lot of the alternative kind of bread making
Corn and
flours, like,
soy and
yeah,
stuff like that, yeah.
yeah,
Okay.
amaranth and teff and all that. And then the array 10 is the much more comprehensive test.
Carolyn,
Yeah.
for that FUT2, now the list of the first 3 I'm going to give you together. When all of them are homozygous together, they are a problem as well. The first 3, they have to be together homozygous. RS492602, RS601338, RS602662. Now, those 3 together homozygous will cause a problem. I guess if one's just homozygous, maybe not, but I know those 3 together. I think Jacqueline Greenfield is more of an expert, but one of us can contact her and see why. Now, the one standalone, the RS1047781, that one just by itself causes a huge problem when it's homozygous.
Yeah, and
Okay.
it's the 601338 that is associated with the issue with the gut bacteria.
Okay, so it's the 601. Okay. Yeah, I know they tend to be low in bifido, right?
Yeah.
Okay.
Okay, just I think this must be related. So Andrew's just saying RS281377 question mark. So if that doesn't make any sense, you might have to be a bit more specific, Andrea. Does that make sense to any of you?
No, I think maybe she's asking if that's the RSID. I haven't got my
I know the RSID number, and I think that's one of the HLA ones. I think that's an HLA one.
Oh, right, okay.
Okay.
I kind of have these memorized. I believe it's an HLA.
Okay, Jennifer is asking Carolyn, if Cyrix testing is not available in Australia, what lab do you recommend for equivalent testing?
Good question. Well, I've actually contacted Cyrix 4 times this year because I'm so desperate to get these here. Unfortunately, we don't have wonderful celiac genes. Our clinic is in the process of trying to get a lot of these HLA genes, particularly when they are associated with mold and Lyme. So we're in the process of working with some labs in trying to get that done. But there's not really great ones, and I'm just really hanging out. Cyrix said that they decided this year to go into Ireland, and they hope to be in Australia early in the new year. So I will be contacting them again in the new year because I've heard Cynthia talk for years about all the Cyrix, and I've looked into them all, and I just really want them. So no, unfortunately, we don't have anything that's comparable.
Okay. Could you give the FUT2 practitioner's name again, please?
Oh, that was Sterling.
Jacqueline Greenfield. She's on our Find a Practitioner list at MTHFR support. She's located under UK, and she also answers a lot of questions on our forum.
She's gone because the dog's barking.
Oh, funny. Okay, so Andrea, just put your hand up. Was your question answered before I ask these other things? Hang on a sec. I'm just going to flick to your screen. Sorry, could you put your hand up for me again if your question was answered? Fantastic. Thank you, Andrea. That's great. Okay, I have another question here. Will new autoimmune report cover mold, HLA, SNPs, aka shoemaker?
Sterling isn't here, but we were talking about this the other day, and I believe yes, it will, because it's going to be a whole 40-page report in its own right. So yes, I believe it will have all those SNPs.
Okay, great.
I'm going to have every HLA that there's peer-reviewed research. Yeah, every HLA gene with peer-reviewed research that is on 23andMe, I am pulling out on this new report we're going to be putting out. And we also have National Geographic and Ancestry.com coming out as well.
And when will it be ready, Sterling?
Well, I've had to hire a few people, so it won't take 2 years and possibly 3 months, 2 to 3 months. Hopefully 2 months, but you know things happen, so it might be 3 months. I'm probably going to have people like you and Cynthia to take a look over it, and you guys might say, hey, add these, and so sometimes I let you guys look over it to see if there's any other autoimmune things I haven't added, and you guys might want a few more things added in. So it might be a little longer than 2 months. I say maybe 3.
Yeah.
Yeah, I just want to mention when Sterling does these reports, not only does she have to identify the SNPs that are relevant, but then she has to go through all the research and pick out the keywords, because on her website you can enter in a word, a disease, lots of things, and with these keywords pull up these searches. And so it's not as easy as just identifying them and putting them on a sheet of paper. You have to do hours and hours of research, and sometimes one RS number will take her all day.
I can vouch for that, because a lot of the research when we've been trying to find whether an allele or a risk allele is an upregulation or downregulation, sometimes I can search for a week. And it's really difficult to find because they don't always in the research say whether it's an upregulation or downregulation. So, yeah, it's very time consuming.
Mm-hmm.
Okay. Now, we don't have any more questions. So is there anything that any of you wish to add to the end of this webinar?
Yeah, I wanted to say one thing. I left this out when we're talking about APOE and fat balance. Part of balancing fats is to make sure that the nutrient cofactors are present for the enzymes that convert the fats. So again, it's the basics. It's your B vitamins, it's your minerals, alpha lipoic acid. It's a good functioning Krebs cycle, because some of the output of the Krebs cycle are cofactors for these enzymes that are converting the fats.
Yeah, good point.
Okay. Well, if there's nothing else to add, then I'm just double checking. We don't have any more questions. So if you do think of any more questions afterwards, please feel free to email. Another great webinar. I thought that was absolutely super. And we have our last webinar on Tuesday of next week at 7:30 for the US, and Wednesday, the 18th of November, at 12:30 for Australia. And that is webinar 11, pulling it all together. So be sure to attend, because that'll obviously be a summary of everything. And I'd like to say thank you to all of our attendees that managed to get onto this webinar today. And once again, thank you very much to Sterling Hill, Cynthia Smith, and Carolyn Ladowski. So thank you, ladies, and we'll see you next week for the final webinar.
Can I
Thank you
just
very
say
much.
one thing? I just wanted to say in regards to next week's presentation, obviously we're going to be putting together what we think are the key issues when looking at these variant reports, taking a full case history. But if you have specific questions that you would really like addressed, and when I mean specific, I don't mean this patient has this, and this, and what should I do about this, but I mean real issues with the variant report, or being able to pull things together, or if you have key concerns or things that you've seen in practice that you seem to get a response to negatively, or you have pathway issues, then please email it to that info@mthfrsupport.com.au in the next, say, 24, 48 hours, and we will try and incorporate those specific questions into the presentation so that we're answering the things that are bothering you the most. That's all I want to say.
Excellent. That sounds great. Good. All right, then. So remember, you just have to email those questions. You see the email there at the bottom of the slide, info@mthfrsupport.com.au. So have a lovely evening and a great afternoon, everyone, and we'll see you next week. Thank you.